Welcome to our channel. There is a botanical mystery currently unfolding in modern cultivation facilities, one that directly challenges our fundamental understanding of plant biology and secondary metabolite production. Across advanced greenhouse canopies and carefully managed outdoor crops, cultivators are observing phenotypes that produce physiological effects far exceeding what their standard THC percentages should biologically allow. For years, our entire agronomic focus has been on pushing the plant to its absolute metabolic limits, maximizing every variable from substrate moisture to vapor pressure deficit in order to drive up that single delta-9 THC number. Yet we are now routinely seeing plants with surprisingly modest THC testing profiles delivering experiences that completely shatter expected potency ceilings. This paradox suggests that our singular obsession with one molecule has blinded us to a much deeper biochemical reality occurring right inside the resin glands of our crops.
To understand why this is happening in our gardens today, we have to look closely at the shift from wild-type genetics to highly hybridized modern cultivars. In its natural evolutionary state, the plant grew with a highly balanced, albeit relatively low, cannabinoid profile designed strictly for natural defense and UV protection. But as cultivation moved into highly controlled environments, growers began aggressively selecting genetics for extreme psychoactivity. We altered the evolutionary trajectory of the plant, forcing it to allocate massive amounts of its daily photosynthetic energy toward producing secondary metabolites. But there is a biological ceiling here. A plant can only hold so much resin by dry weight before the physical stress causes the stalk and cellular structure to completely collapse under its own mass. If modern flower has largely hit its maximum carrying capacity for standard THC biomass, why do current limits fail to account for these seemingly extreme, unprecedented highs we are seeing in the latest harvests?
The raw numbers illustrate an agronomic shift that is nothing short of an agricultural anomaly. Older landrace genetics traditionally capped out naturally at single-digit psychoactive percentages. But today, driven by intense selective breeding and aggressive crop steering, it is not uncommon to see modern phenotypes routinely testing between 25 and 30%. To put that into perspective for a grower, this means nearly a third of the dried flower's total biomass is comprised entirely of active secondary metabolites. Achieving this requires immense environmental precision. It means pushing light intensities to their absolute maximums, tightly controlling aggressive root zone drybacks, and managing canopy temperatures down to the exact degree to mimic late-season stress. But this mathematical climb in THC percentage only explains a fraction of the modern experience.
To truly grasp this fundamental shift in our canopies, we first need to re-examine an agricultural technique that completely rewired the plant's biological imperatives.
This was the Sinsemilla revolution. Cultivators learned that by meticulously isolating unpollinated female plants, they could entirely prevent seed development. Biologically, a female plant's primary goal is reproduction. When denied pollen, she becomes increasingly desperate, forcing all of her metabolic energy heavily into the development of sticky, aromatic secondary metabolites in a final attempt to catch airborne pollen. This forced biological frustration dramatically increased the sheer volume of resin produced per 1 square meter, or roughly 10.7 square feet, of canopy space. The highest concentrations of these active compounds are localized within the dense bud clusters known as colas. Within these colas, you have the pistils and stigmas acting as the reproductive organs, entirely surrounded by tear-shaped bracts. These bracts, along with the protective calyxes at the base, are the primary real estate for trichomes. When a grower introduces strategic environmental stressors, like dropping late-stage flowering temperatures to 15 degrees Celsius or 59 degrees Fahrenheit, these microscopic mushroom-shaped resin glands swell with complex oils, producing a vast array of trace phytocannabinoids that we are only just beginning to map out. Because of this, cultivators today do not just grow for raw yield anymore. They grow for specific resin traits that dictate which extraction path will yield the best final product. How a grower manages the living plant directly impacts trichome fragility, terpene retention, and overall glandular maturity. For instance, specific genetics grown under precise vapor pressure deficits yield robust resin heads ideal for solventless methods. Using exact heat and immense mechanical pressure yields sap-like rosin that preserves the plant's delicate terpene profile directly from the field. Conversely, other phenotypes are deliberately cultivated for chemical isolation, where closed-loop butane extraction can strip the biomass to yield glass-like shatter reaching up to 90% THC, or molecular distillation can strip everything down to a 95% clear, viscous fluid. This hyper-focus on glandular output proved that we could push the final product far beyond the biological limits of the raw flower itself.
And this brings us to a rather profound realization about modern cultivation.
The gap between a 1980 joint and a 2024 one is not incremental, it is entirely categorical. But it is crucial to recognize that this leap is heavily driven by advanced agronomy rather than just cultural history. We're utilizing highly controlled greenhouse environments, deploying precise full-spectrum LED lighting, and heavily advanced substrate dynamics to push the physiological limits of the plant. We have engineered microclimates that provide the exact spectrum of light needed to maximize resin gland expansion, paired with precise irrigation strategies that fuel secondary metabolite synthesis at rates that are utterly impossible in unmanaged nature.
Yet, despite all this agricultural mastery and the undeniable rise in raw potency percentages, a massive blind spot remains in our understanding of the plant's effects. We must now address the pervasive myth that standard delta-9 THC is the sole driver behind these unprecedented physiological highs. The assumption that simply increasing delta-9 levels equates to a linearly stronger experience completely ignores the vast, hidden complexity of the plant's internal chemistry. There is a hidden variable in the modern resin gland, a molecule synthesized in such trace amounts that it remained entirely invisible to standard laboratory equipment for decades.
Please like and subscribe, it really helps our channel a lot.
The answer to our opening botanical mystery was finally revealed in 2019 by a team of Italian researchers analyzing a specific medicinal cannabis variety known as FM2.
Using highly advanced mass spectrometry, they discovered a novel, naturally occurring phytocannabinoid that had never been documented in the history of cannabis science. They named it tetrahydrocannabinol, or THCP. This wasn't some synthetic creation made in a lab. This was a trace molecule naturally synthesized by the plant itself, hiding in plain sight within the trichome heads of specific genetic lineages. The complex molecular biology behind this new molecule explains exactly why certain phenotypes feel overwhelmingly potent despite average test results. THCP is almost identical to standard THC, but with one critical architectural difference. The resorcinol moiety of the THCP molecule features an extended alkyl side chain. In pharmacology, the length of this specific carbon chain acts as the primary pharmacophore, meaning it dictates exactly how tightly the molecule can bind to the human endocannabinoid receptors.
To make the biochemical mechanics behind its extreme potency immediately digestible, let us contrast the two structures. Standard delta-9 THC possesses a 5-carbon alkyl side chain. It fits into our receptors well enough to cause the traditional psychoactive effects we expect. But THCP possesses a 7-carbon side chain. Those two extra carbon atoms change absolutely everything. Because of this extended physical structure, THCP fits into the deep hydrophobic pocket of the human CB1 receptor with an astonishing 33 times greater binding affinity than standard delta-9 THC. They allow the molecule to anchor itself so deeply and securely into the receptor that the physiological response is drastically amplified. This 7-carbon structure means that even a fraction of a milligram of THCP produced naturally by a well-grown plant can influence the human body far more aggressively than massive amounts of standard 5-carbon THC.
But here is where we must pivot to the practical cultivation reality of this newly discovered compound, because the difference between what the plant produces and what the market sells is staggering.
While THCP is incredibly powerful, nature produces it in truly microscopic quantities. In the original plant samples, it was found at concentrations of roughly 0.01%. To extract just 1 single gram of pure, naturally occurring THCP, an extractor would need to process between 750 and 5,000 grams, or roughly 1.6 to 11 pounds of premium raw cannabis flower. The agricultural economics of naturally sourcing this molecule on a commercial scale simply do not work. Because natural botanical scarcity makes genuine THCP extraction financially impossible for mass-market products, the industry has been flooded with a red herring. The vast majority of commercial THCP products available today are actually semi-synthetic. Manufacturers take readily available, inexpensive CBD isolated from industrial hemp and subject it to aggressive chemical catalysts to synthesize THCP in a laboratory. They then take this lab-created distillate and spray it directly onto low-THC industrial hemp flower to mimic the look and feel of natural high-potency genetics. For a dedicated cultivator who spends months perfecting substrate biology and environmental controls, this practice of coating inferior biomass with chemically converted isolates represents a significant disruption to product integrity and agricultural transparency.
And as you might expect, these influx of semi-synthetic isomers has completely destabilized how we measure and verify potency.
This flood of highly potent molecules has subsequently triggered a severe analytical crisis for testing laboratories. The infrastructure designed to ensure consumer safety and accurate agricultural grading was built almost exclusively to measure the standard 5-carbon cannabinoids. When cultivators or manufacturers submit samples containing high levels of these novel 7-carbon isomers, standard testing equipment often fails spectacularly. The specific testing hurdles are immense. First is the molecular mimic problem. THCP and its synthetic variations share nearly identical molecular weights and structures with other cannabinoids. When run through traditional gas chromatography, the intense heat required by the machine can alter or completely misidentify the compounds. This leads to coelution errors, meaning THCP bleeds directly into the data spikes for delta-8 or delta-9 THC on the test graph, resulting in wildly inaccurate potency scores on the final certificate of analysis. The only reliable solution for laboratories is to upgrade to high-performance liquid chromatography, or HPLC. This method operates without destructive heat, using liquid solvents and high pressure to physically separate the molecules through a column at room temperature before they are measured, finally giving growers an accurate view of their crop's true chemical profile. Which leads us to a fascinating conclusion regarding our canopies. If single-digit trace elements can rewrite the entire human experience of the plant, what other microscopic molecules are currently lying dormant in your canopy, just waiting for the right environmental stress to wake them up?
If you want the full uncut data and this week's newest plant science papers, head over to the members area. You can tap into the entire Cannabis Science Brain Vault for just 99 cents a month.
Thanks for watching. This content does not constitute medical advice, legal advice, or recommendations for the use, self-treatment, or consumption of any substance.